Thank you all for coming to this last session. Hopefully, we'll make it worthwhile here. And I'll get started in the interest of time. And our objective here is the goal of Cellerant Therapeutics is to transform lives by harnessing the power of stem cells, hematopoietic stem cells specifically, to develop and deliver immunotherapies, innovative immunotherapies for treatment of blood cancers and blood-related disorders. Now, among all blood cancers, AML is probably one of the biggest one with a huge unmet medical need. And I don't want to go through all of the statistics here, but it's suffice to say that about 10,000 of these patients die every year. The incidence is about 20,000. The median age is 68 years, and the 5-year survival rate of patients over 68 is about 7%. And the reason for this poor prognosis of these patients are twofold. One is the current chemotherapies that are being used are extremely toxic. That is daunorubicin cytarabine, as it's the common name for that is 7+3 induction therapy. It's extremely toxic and it causes significant infections in these patients. Not to mention when these patients relapse or become refractory, they get more chemotherapy and the infections get worse, and many people die of infections. About half of the patients over 68 refuse to take any treatment because there's a potential of them surviving longer without taking treatment than going through some of this treatment. So our first product that I'll talk about today, we just completed phase 2 clinical trial, is focused on addressing these infections. So why are there so many infections in these patients? Because the chemotherapy non-specifically kills all of the circulating neutrophils and the progenitor cells, because that's what those chemotherapy drugs are aiming at doing, because leukemic cells are in circulation. So what we provide is innate immunity, and I'll talk more about that. And then the second aspect that we're going to address today is the relapse. Why do they have such a high rate of relapse? That's because chemotherapy does not kill the leukemic stem cells, the stem cells that survive after chemotherapy treatment and come back to form more tumor cells. We're going to address both of them today in our presentation. So it's a clear unmet medical need, and hopefully with these products we are able to treat AML better. So the first one, what we call as CLT-008, is an allogeneic myeloid progenitor cell product. So we get cells from normal healthy volunteers. These are hematopoietic stem cells, what's called as CD34 cells. We expand them over certain days. We pull the product from multiple donors, and that's available as and when it's used, when it's required. It's a frozen product. So this product can produce neutrophils in vivo to fight bacterial and fungal infection. And I'll show you phase 2 data that has clearly demonstrated significant reduction in infections, days in hospital, and use of antimicrobials in these patients. And we are now poised to get into a pivotal phase 3 trial sometime in 2019, Q3 of 2019. And the market opportunity is significant, not just in AML, even in other indications where the neutropenia is prolonged. As you all know, G-CSF is an approved drug for neutropenia for many, many indications, and it works very well when there are progenitor cells present. In the absence of these progenitor cells, which will happen when they receive myelosuppressive drugs, the G-CSF has nothing to work on. As I'll show you today, we are going to compare side by side with G-CSF in our clinical trial. The second program is an antibody drug conjugate. In other words, it's an antibody targeting leukemic stem cells, targeting CLL1 expressed on leukemic stem cells, to which we attach a cytotoxic payload. Once the antibody drug conjugate goes inside the cell, the payload gets released and the cell gets killed. We'll talk about that. That program is moving towards an IND filing in Q1 of next year. So in terms of 008 product, let me describe a little bit more. It's an off-the-shelf product. It is not patient-specific. It's available as and when required. It does not match to the recipient, and it is very user-friendly. It is thawed by the bedside and given to the patients as and when required, and it's a simple IV infusion. There's no detailed processing required at this point. We have now dosed over 150 patients in our safety trials, in our 3 clinical trials so far. So our randomized phase 2 open-label control trial involved 78 de novo patients receiving 7+3 induction therapy. There was another arm in this trial of another 42 patients who received high-dose chemotherapy, which is higher version of higher concentration of 7+3, but I won't get into that today because our focus for phase 3 is going to be on 7+3, as it is the largest induction chemotherapy used in this country and outside of US as well. So we tested our product on these patients. They received the product on day 9, 10, or 11. Why that time frame. First 7 days is when they get chemo. And then we wait for 2 days before we give our product. So the product is given on 9, 10, or 11. And then we give GCSF starting on day 14 because day 14 they do a bone marrow biopsy. So the physicians like to give GCSF only after they complete that. So it's given on day 14. The control group also receives GCSF on day 14. These patients will receive any antimicrobial as decided by the physician for the treatment of infections, antibiotics and antifungal agents. There are no restrictions to that in this trial. And infections were adjudicated by independent blinded experts who were not told which was the control of the treatment group. And patients were followed up to 42 days. What I'm going to show you now is the data from infections, decrease in hospital days, and also use of antimicrobials. So we saw a significant decrease in the use of in infections in these patients. It was a 73% decrease in infections between the control and the treatment arm with a p-value of 0.0098. So this was infections between day 15 and day 28. That's after we give GCSF through day 28. So this was very clear from this data. And what I don't mention here is if you look at these patients from day 21 to day 42, 21 is when the neutrophils come back in all of these patients. But even then, because the residual infection was not eliminated because they didn't have their own immunity, these patients 21 to 42 in control group had 10 infections compared to 0 infections in the treatment group. So these cells do two things. One, they therapeutically address the infection, and secondly, they prevent infection and also remove any of the residual infections that are present. And we also saw 3 days decrease in hospital stay. And this 3 days is because of the lack of use of IV antibiotics in the treatment arm. They didn't have to go through that significant treatment. And obviously, as you would expect, with the decrease in infections, we also saw decrease in use of antimicrobials in 2 different scenarios. One is when they intensify the antimicrobial use because of fever. As you can imagine, these patients are extremely neutropenic, which means they don't have any neutrophil counts in their body. When the fever goes up, they intensify the antimicrobials. 43% of the patients, it was significantly reduced in the treatment arm. And 44% of the patients in the treatment arm, or there was a difference in 44% between the control and treatment arm, who did not have to receive dry antimicrobials for the treatment of infections that were diagnosed. So essentially, the cells did the job of what they needed to do. It was a significantly decreased use of antimicrobials. There were no safety issues between in the treatment arm, and SAEs were quite balanced, except for infection-related SAEs that were more in the control arm compared to the treatment arm. These are the serious adverse events this patient's experience. So, the market is significantly large. I just talked about AML, but then there's clearly a potential for other malignancies, heme malignancies, and as well for 5 to 10% of solid tumor patients who receive myelosuppressive therapy. So I'll quickly summarize this before I go on to the next program, and I'll just probably focus on the last 2 bullet points. We have an end of phase 2 meeting planned with the FDA in June of this year, and we also plan to initiate our phase 3 clinical trial Q3 of next year. So with that, I will move on to the antibody drug conjugate program. Here, as I mentioned to you before, the target is CLL1. The more well-known target for AML patients is CD33. There's one drug that's in the market right now called Mylotarg, and there was another drug in development that was stopped last year in phase 3. That is CD33 ADC from Seattle Genetics, and that was stopped because of infections in patients and related death towards infections. So what we are going to show you here is the effectiveness of this product in different models. Again, it's a very potent molecule. This is an antibody drug conjugate. As I mentioned before, antibody binds to the cell. The payload gets released inside the cells and kills the cell. We see very nice efficacy here. And then we also see significant safety that this particular drug demonstrates. What this graph shows you here is we take human hematopoietic stem cells, put them in mouse models, and actually see whether they can differentiate in normal blood cell types. What you see, the first 2 bars there, the control group and our treatment group at ADCLT030, there is no decrease in the type of cells they decrease. But if you use CD33, you see a significant decrease in the differentiation of the normal cells, which is why you see increase in infections and death when you use CD33 ADC. So with that, I am going to conclude this part of the talk, which is 030, And again, it's a wonderful target that is has a better safety profile than CD33. And our initial plan is to go into patient population that I discussed before, where they decline intensive chemotherapy. So we would start with the monotherapy. And our target filing, IND filing, is Q1 2019, and soon after we'll initiate our clinical trial. In the interest of time and the fact that we're going to ask some questions right at this point, we're not going to summarize too much, but our key goal is to seek capital to fund our phase 3 trial for 008 and do a phase 1/2 for 030. Thank you.
Great. So, Ram, my first question for you is, based on the results from the phase 2 study for 008, how does that inform the design of the phase 3? And tell us what you think that's going to look like at this stage.
Sure. So our phase 2 design involved, as I mentioned to you, de novo AML patients 55 years and older. So we plan to stay with the same patient population in our phase 3 as well. These would be patients who would receive 7+3 induction therapy, de novo AML patients over 55 years old. So we are going to stay with the same patient population because there's a big unmet medical need in those patients. So if we can get them through the first stage of chemo and then get them to go through complete remission, that would be ideal. So we are going to stay with the same patient population. So design for phase 3 would be 1-to-1 randomization. Again, serious infection would be the primary endpoint. Serious is defined by, when I say serious infection, it's defined by the use of IV antimicrobials. That's how we're going to define serious infection. That's an accepted definition from the FDA as well. So that's how the design is going to be. We expect about 230 patients total between the two arms.
Okay, and you're hoping to commence that study in the second half of this year?
No, it will be our meeting with the FDA is planned for June of this year. It'll be in the second half of next year is when we will be able to, because it takes time to generate the product and things like that.
Okay. With respect to your ADC, again, as you mentioned, slightly different target, but what else about the payload itself or the linker itself that could enhance the safety and efficacy of this approach?
It's a very good point. The drug that's out in the market right now, Mylotarg, the reason why it's so toxic, besides being expressed on normal stem cells, is that the linker is not stable. It falls off. So it was on the market. It was taken off the market because of toxicity. They fractionated the dose. They went back in and got an approval. So in our case, the linker is extremely stable. It doesn't fall off in the plasma. And second, the payload is a DNA damaging payload, which is shown to be extremely effective in an AML setting where you have a number of cells that are quiescent, that are not cycling. So if you use a DNA damaging payload, it's more effective in killing. So it's a very stable linker payload combination.
And you also have the ability to have this as an application for acute radiation syndrome as a counterterrorism measure. BARDA certainly is interested in that and has provided funding. What do you need to do to complete that program?
Sure. So this is going back to the CLT-008 program, the myeloid progenitor cell program. So we have application in AML and, as I said, in other cancers, but also a large application in treating acute radiation syndrome, which is the type of neutropenia that's caused when exposed to nuclear accidents or some form of radiation. So there, what we need to show is we fall under what's called an animal rule guidance, where you have to demonstrate efficacy in 2 animal models and safety in humans. So the 2 animal models we have to demonstrate would be mouse and non-human primates, and that's ongoing. That's funded through BARDA. And the safety is demonstrated to the AML clinical trial, which is also funded through BARDA. So the complete CLT-008 program currently is funded by BARDA.
Okay. Well, very good. Thanks so much.
Thank you.